Oh, hey there. Someone said I needed to start giving shout outs. Like, shout out to my first period. I don't know. I don't understand. I don't understand how it works. You can explain to me how shout outs work. And then you can explain to me why. There it is. Aha, the PowerPoint isn't working. Okay. Topic 1.5, folks. Topic 1.5 is kind of a what do I want to say? It's different from everything else we've done in topic 1, meaning that it is more theory based, and we're talking about the origin of cells, right? The history of cells, but not so much the history, because this is one of those epitomes of a model where we have to look back at the evidence that we have and what we can recreate today. But remember, at the beginning of the year, we were talking about the buckets. Like, models are things that happened too long ago, or too far away, too small, too big, things like that. So when we're talking about the origin of cells, the things that you really need to focus on are the evidence that we have to support this model of cell origin, the gaps in the knowledge, and then some current experiments that have been done to try and recreate these, right? And why they haven't worked. So kind of focus on those things as we go through the origin of cells. Okay. Here's kind of a topic outline. Put this up here, kind of move this around. Looking at non-living synthesis. So synthesis meaning to make. We're going to again look at these two experiments by Miller-Urey and then Louis Pasteur, who you probably have heard of. We're going to talk about how the theories of not only like the first cells, but prokaryotic cells, and then how DNA is changed, and then how oxygen plays a factor in creation of life. Okay, so that's the things. It's a pretty long lecture. I'll try and get through it as fast as I can. Lots of information on the slide, so make sure that you're pausing it, writing it down, and then kind of listening to the story. More of like a story-based runs. Okay. First of all, let's go back and look at a good review of one of our characteristics of life, which is metabolic reactions, right? Homeostasis, being able to maintain an internal environment that is different from the environment around us, right? So the primary function of cells is to create this internal environment that reacts to the outside world so that it maintains the same inside, right? So for instance, it gets too hot outside, right? Our cells function, our body's function is like to cool us down. It's too hot inside our body, like a fever. Its function is to fight against it, like water in, water out, energy in, energy out. We are to maintain an internal environment that is separate from the environment around us. Okay. Now, those metabolic reactions that we'll talk about a little bit later into like energy and mitochondria and all that kind of stuff, those are absolutely vital to sustain life, to keep life going, right? So we have a cell, it's made, and now it needs to be able to keep living, right? This, one of the things that allows it to keep living, right? The organism to keep living is its ability to self-replicate, right? So we've talked about how cells, whether they're prokaryotic or eukaryotic, are able to replicate, right? And that's actually part of the cell theory, that all cells come from pre-existing cells. So without us doing anything about it, our skin cells are going to replicate and replace themselves. Our blood cells, our body cells, all these cells inside of our body are going to automatically just double when they need to, right? That's the capacity for self-replication. So basically, that says at the bottom, is like a cell, what defines it is its ability to maintain it separate from the environment around it and to replicate without like being told to. Okay, metabolism and homeostasis and self-replication are all one of those characteristics of life that we've gone over in 1.1 that you will still be responsible for knowing. Okay, now the theories on how, so if all cells come from pre-existing cells, well, that kind of creates a problem, right? It's kind of like chicken or the egg that comes later in the lecture. Like, what came first? And so there's a couple different theories on how those things came to be. This is our first one, talking about like the first origin of cells, right? So how did we first get these, the molecules that are necessary to be part of cells, right? It's going back to your bio that were, carbohydrates, proteins, nucleic acids, lipids, those types of basic carbon-based molecules are essential in our cells, all right? And so here's how it goes. I was thinking that as you're taking notes, so this one goes into 1, 2, 3, 4 order. Some of them don't, so make it more of like a story, like this happened this, and this happened this, kind of like this one right here, right? Draw this diagram into the margin here, right? So here's what it says from non-living material. There was non-living synthesis of organic monomers, right? Single, mono meaning one. So I've got this inorganic compound and they assembled into complex polymers, right? Inorganic means they don't contain carbon, and then all of a sudden they do. So they create these rings of carbon, and they've got these different organic monomers, like I said, nucleic acids and sugars, right? And the basic building blocks of what makes our complex. Now, as these were built in the complex monomers, the monomers kind of came together and started self-replicating, right? So they created these bonds. Again, this is just like the theory. We're going to look at evidence for and against. They created these bonds, and they became able to, we know this bond is, I want to say, kind of happens naturally, and so they start replicating. And then those molecules became somehow enclosed in a membrane. Remember, our membranes are made of lipids. Lipids are fats, which are just other organic or carbon-based molecules, right? So we'll talk about those a little bit later, right? So that is the formation of non-living cells. So like how that went about. So if you had to say, what is the like 3 steps or 4 steps of how the cell formation came from non-living cells, we talk about inorganic to organic to like, so simple to complex, and then being packaged into membranes that creates what we call the bionets that gave rise to simple cells. So this is my first type of molecule with my membrane around it, and I've got my polymers connected here, protobiontic formation, right? So this is like pre-cell. Then comes, right? So then how do those like all come together, right, to create a living thing? So the Miller-Urey experiment was called, he was looking at what might have existed at the beginning of Earth, right, when under all these different, what I'm going to say, like temperatures, things in the water. He called it the primordial soup, right? So he literally called it the primordial soup, like the beginning. And so he wanted to show how situations on Earth at that time, so heat, Earth is covered in oceans, you still have like very volatile, you still have things hitting the Earth, you still have what's it called, volcanoes. This is obviously sign language for volcanoes. So he's trying to recreate those situations because our Earth is not like that anymore. So here's what he did, right? His primordial soup. The water was boiled to kind of like as a story. Water is boiled at a very high temperature, and it's allowed in a reducing atmosphere. So take here, ready? Woo-hoo. So I've got my heated water here, right? And then it's going up here with the gas. So it's allowed to escape here. It is then exposed, this atmosphere is exposed to an electrical discharge, like lightning, trying to kind of show like how lightning striking the air could, like what the effects of that would be, right? With this and this oceans evaporating, coming up here, air, now it's electricity, right? And then it was cooled and allowed to condense. And what he found is that by that happening, with all those like monomers in there, those very simple compounds going up into the atmosphere with electricity, that condensed mixture contained organic molecules, right? So all of a sudden we're carbon-based. So he was able to recreate that in a laboratory setting, right? Trying to show like what early Earth was like and some situations that might have occurred, right? So how did these things come about? So Miller-Urey, I'm sorry, there's 2 of them. I kept saying he. Those 2 were able to do that together. And this is kind of basic of their experiment, and the results from that was that they were able to create that with those conditions. Okay, so what happened? So why did they use those things? So the boiling water, the hot water, the evaporation, and then the electricity in the air. Like what were they trying to recreate? So what we know is that it has to have these environmental conditions. So no oxygen in the air, and then an electrical discharge or heat of some type being exposed to that atmosphere. Now, we still see these 2 situations in places on our Earth around hydrothermal vents and then active volcanoes. And so there's thought to be that those were the source of heat. If you go like history of Earth, like as it was recreating, volcanoes erupting, Earth cooling, right? Still very volatile. Earth mostly covered in water and very hot, right? Because we don't have any atmosphere to regulate our temperatures. And so a couple different locations that would exist right here. And then one that we don't have evidence of, but is theorized to, are comets coming in. So comets bringing water, comets from, going around the solar system, hitting the Earth and bringing compounds that were not currently present on Earth. Okay. All right. So that's kind of like, okay, how these just molecules came to be, those non-living. All right, so, but how do we show that living things can then create from there? So Louis Pasteur, who 1800s, pretty famous scientist, and there's actually glassware named after him that you'll see here in a little bit, so like Pasteur pipette, things like that. So he was trying to create the conditions for the non-living synthesis, because those don't exist on Earth anymore, right? We don't have that same type of Earth. So he said living cells cannot just arise independently by, it's called abiogenesis, right? So he was trying to prove that cells can only come from pre-existing cells, and the law of biogenesis is largely attributed to his work, right? So here's how biogenesis works. Okay, so Louis Pasteur, there he is. All right, what he did. So again, kind of explain the methodology in that. So he took, and your book has a little bit better I want to say visual of what this looks like. But Pasteur's experiment basically went like this. He was trying to show that it has to be exposed to the atmosphere for anything to be able to come in and life to grow. So whether it was mold and fungus. So he took a broth that contains like all of the energy that's needed, like the fuel that's needed for things to grow, and he boiled it to kill anything that was in them. So he sterilized the environment. Now, he melted glass so that it did this little like, dip here, right? So it goes down like this, so that as it heated and evaporated, what happened was it created a natural seal, right? So I get this water that's blocking the exposure to the outside atmosphere that has all these germs in it. And so what he found is that as the broth cooled and the water collected, because it sealed that exposure, there was no growth in his soup here. Soup, his broth, right, that contained all that those nutrients. However, the minute it was exposed to the air, right, so the air was able to come in, that has a lot of microorganisms into the broth, there was instant contamination, right? So I get mold, bacteria, things growing within a week, right? And so again, we'll look at this in your book because it shows it a little bit better. But what he showed is that there has to be exposure to the atmosphere, things that are coming in, right, in order for any type of life to grow in an area. So if it's blocked off, right, whether it's a closed system or with this encased like a water barrier, there's no growth that occurs. All right, so exposure to air. All right, going back to that. So those are the two famous experiments. Chicken or eggs. All right. So replication of cells requires those instructions in your DNA, but you have to have the DNA to have the instructions, right? It's like what came first, the chicken or the egg, right? And so what we do know about DNA, and if you're, back in bio, and then we'll talk about later, is that it is the genetic code with all the instructions for everything, right? So those are the instructions, but what they create the instructions for are proteins, and the proteins are the things that do all the work, right? So the DNA is just like the directions here, go make the thing, right? The proteins are made. It's the ribosomes are the chefs, and they make the proteins, okay? However, proteins require the DNA to be made, right? And the DNA requires proteins in order to self-replicate. So like they depend on each other to both exist, right? To function, right? Proteins require DNA, but DNA requires the protein in order to round them on itself. So the conundrum is like, what came first? What molecule came first, the DNA or the protein? That's kind of the still like, when I say things that we're looking to for more information on, right? Which molecules came first, origin of cells, protein or DNA, right? And so scientists have looked to something called RNA, and this is now called the RNA World Hypothesis, to try and kind of like help with this conundrum, right? This problem, right? So deoxyribonucleic acid, right? DEoxy, that's where we get our without oxygen. That is where we get our DNA. That's the acronym for DNA, right? RNA is just ribonucleic acid, right? So ribonucleic acid, it has doesn't have the deoxy in front of us. RNA is able to self-replicate, right? Making it like an effective coding molecule. So it's able to just self-replicate, and it can actually causes a catalyst, which is something that starts a reaction in chemical reactions, right? So this little molecule has different abilities than DNA does, right? And because it's been superseded by DNA and proteins, right? So that means it came before. So the things that came after DNA, they're more chemically stable because they've got RNA is single-stranded too. Sorry, I forgot that little part there. So it's more chemically stable because it's double-stranded and it's twisted together, so it better codes for molecules. And the proteins that are made from those have greater variability because they have ability to make 20 amino acids, so they're able to do more, better catalytic molecules. So RNA is thought to be that kind of transition from DNA and protein, right? So it was like the original molecule that was able to do those things, and then DNA and proteins came about from RNA, right? So RNA, DNA, proteins at the same time. So that's called the RNA World Hypothesis. So where did DNA come from? Chicken or egg? This is one of the ideas that explains that. Okay, so that's all non-living stuff. Sorry, I just feel like this unit's like huge. So that's all the non-living stuff. So now let's talk about, okay, so we have all that, those basic things that we need. We've got DNA, we have lipids, we have carbon base, right? Molecules, all those organic. And now let's talk about, so how did we go from these very basic prokaryotic cells to like eukaryotic, right? So how did that happen? And here's the theory behind this. It's called endosymbiosis, right? Endo, which is a review from your last one, like to go into. Symbiosis means to like exist together. So they like engulfed each other and became one thing, okay? And so here's how that goes. It's that prokaryotes, right? So very ancestral prokaryotes, super basic, right? I get this kind of area where I find that the nucleus starts to form, and then they became engulfing kind of each other and then didn't digest. So kind of just like almost like became part of it, and then gave that new cell a new function. So I have this like anaerobic bacterium, right? That looks like my mitochondria. This cyanobacterium, this looks kind of like my chloroplast, right? I'm able to make like light, photosynthetic. And so those are just kind of engulfed by the cell. And now all of a sudden I have organelles. So those different types of cells came together to make organelles that had different functions and different things for the cells, right? So this is kind of our ancestral eukaryotic cell. It shows that I get the DNA starts to form in the middle. They're aerobic, right? So requiring oxygen, mitochondria, the cyanobacterium comes together, and I have my basic cell. Okay, here's our evidence of that. You can remember that's matter. It sounds kind of crazy. I don't know, mad scientists, right? So evidence of symbiosis, you can write down these 5 things. So one of the first ones is that they both have double membrane. So the double membrane, remember, is supposed to allow things to go in and out. Like that's the function of the membrane. So the mitochondria and the chloroplast both have those. So they're double membrane organelles, but they're within another membrane, right? So that's one of the pieces of evidence is they must have existed separately, that like they were their own thing, and then they came together. Okay. The other thing is that they're both susceptible to antibiotics, which are specific protein, or I'm sorry, that attach to certain proteins that are going to kill off that certain part, right? So it's not just the whole cell; they're both individually susceptible to those. So like they're kind of their own thing. They both reproduce by a fission-like process, right? So just splitting apart, right? Remember we talked about that binary fission. That's how the mitochondria and the chloroplasts divide, and they have their own DNA, right? So mitochondrial DNA, they contain their own DNA. No other organelles have their own DNA. There's like the nucleus has the DNA, but mitochondria and chloroplasts contain these circular, naked, right? So not protected DNA. And going back to what we knew about that little factoid about ribosomes is the 70S ribosomes, right? So all those 2 organelles have those. And so that's the kind of evidence that those were the basic formation of the first eukaryotic cell. Okay. All right. Very last slide here, I promise, is looking at the oxygen-rich enriched environment, right? So how did oxygen come about and create the new situations for life? So photosynthesis is the process that has changed like our atmosphere. So photosynthesis starts to come about, right, in that cyanobacterium, and it's the only significant source of oxygen production on Earth, right? Because it takes in the carbon dioxide, creating those greenhouse gases, and it gives off the oxygen and thus allows other life to come. So the evolution of photosynthetic cells is what changed the composition of the atmosphere and then allowed other types of life to start evolving. So what happened then, when I get this more oxygen, my oceans used to have a lot of dissolved iron, right? Because there's a lot of rocks being, created, cooling off, coming from the inside of the earth, right, released. And that oxygen reacted with the iron to form these insoluble deposits. So they're like banded iron formations. You can actually see them on the ocean floor when you're looking at the polarity of the earth changing. We can see when the poles actually switched because the iron bands, and it'll all automatically align to north and south. Okay, so when the iron and the oxygen was completely consumed, right? So because it was formed with all the iron, it took the iron out, then oxygen started to accumulate in the air. And that's what happened. It goes from an oxygen free to about 20% oxygen, which is where we are about now. And because those oxygen was able to go into the atmosphere, this just feels so long. O3 is ozone, and that started to make that ozone layer around the earth that is super, super small, right? However, our ozone absorbs harmful rays. So anything that was living in the ocean can now come onto land because our ozone has now started to take in all of those really harmful UV rays and the ultraviolet, and anything that would have killed life off on land is now being absorbed by my ozone, and that allows life to now exist on land, right? So you can see how those things progress. Oxygen, iron come out. Oxygen can now go into the atmosphere. Because it's in the atmosphere, it creates ozone, and now life can come out of the ocean and onto land. Okay, here's how what I just kind of explained, looking at key events, right? So first cells, photosynthesis, aerobes, which are things that created their own, plants, and then animals, right? So looking at a billion years ago, and then also looking at my atmospheric oxygen. Go ahead and sketch that into your margins, okay? And this is basically everything I just said with the formation of the ozone, and allows life to change and survival of the fittest and all those things that we've talked about before, okay? And that, my friends, is the end of 1.5. Can you answer the following questions? Can you outline non-living cells and how they came? What did those two experiments add to what we know? What the heck is RNA, and how does that add to our hypothesis of things becoming more complex? How did eukaryotes and endosymbiosis, right? How did that work? And then what happened with the change in oxygen, and how did that contribute to the earth changing? And that's it. It's that simple. It's just, as simple as that. Miss Meyer should probably break this up into 2 lectures. I'm going to think about it. Okay. I'll see you tomorrow. Have a fabulous, or later if you're taking this during intervention. Have a good day, guys. I miss you. It's alone. I'm alone in my room, just talking to myself.